Assalamualaikum friends, I am here Sayyida OMAMA SHAH, and today I am here with a new video in which I will be discussing the mechanism of action of different protein synthesis inhibitor. For that I have a trick, and that trick is buy at 30 sell 50. So from it I took two drugs, and that are aminoglycosides and tetracyclines. So the aminoglycosides and the tetracycline will target the 30 ribosomal subunit, and in this way they will inhibit the protein synthesis. Whereas from cell, the drugs are chloramphenicol, erythromycin, lincosamide and linezolid. Remember over here, linezolid is an oxazolidone. Now these all drugs will target, or it will inhibit the protein synthesis by targeting the 50 ribosomal subunit. So up till now we just studied that the aminoglycoside and tetracycline will act on 30 ribosomal subunit and will inhibit the protein synthesis, whereas the chloramphenicol, erythromycin, which is a macrolide, lincosamide and linezolid will act on 50 ribosomal subunit, and in this way they will inhibit the protein synthesis. Now moving onwards, we have here the initiation complex. Now what an initiation complex is, it is basically the combination of mRNA, amino acyl tRNA and ribosomal subunit. When they all are combined, they form initiation complex. So the drug name oxazolidone, which include linezolid and tedizolid, they will inhibit the formation of initiation complex, right? Okay, now we have translocation. What the translocation is? Translocation is basically the movement of ribosome along mRNA from 5' to 3' direction. So the enzymes, the drugs clindamycin and macrolides will inhibit the translocation. Okay, now moving onwards, we have an enzyme called peptidyl transferase. Now what a peptidyl transferase is? Peptidyl transferase is an enzyme, and it shifts the chain of amino acid from tRNA to from first tRNA and from P side to the other tRNA, forming polypeptide chain, and it binds the two amino acid, forming peptide bond. Okay, so the drug named chloramphenicol, it inhibits the peptidyl transferase. Now I'm going to show you how you're going to remember these all means the drugs that inhibit protein synthesis by showing their different mechanism. So if first I talk about the chloramphenicol, so chloramphenicol is the only drug that contain the alphabet P, and so from here P, I just get the point that this chloramphenicol will inhibit the peptidyl transferase. It's because the peptidyl transferase first alphabet is also P. Okay, now translocation. For translocation we have two drugs, and that are clindamycin and macrolides. So from clindamycin I just get the point cling. So the ribosomes are cling to the mRNA. When the ribosomes are cling to the mRNA, then the ribosome will be unable to move. So if there is no movement, then it means that they will inhibit the translocation. Okay, now the other drug that macrolides, so from macrolide I just get the point movement, right? So it inhibits the movement of ribosome along mRNA, or it prevents the movement of ribosome along mRNA. So the movement of ribosome along mRNA, we studied that it is called as the translocation. So the macrolides and clindamycin in this way inhibit the translocation. Okay, now coming onwards towards oxazolidone, or you can say linezolid and tedizolid. So for that the trick is a little bit complex. For example, I know that aminoglycoside, it is also used aminoglycoside is also used to prevent the formation of initiation complex. So from L I just took like aminoglycoside, and in aminoglycoside there are alphabets LID. So like aminoglycoside, linezolid also is used to inhibit the formation of initiation complex, and aminoglycoside are used to cause misreading of mRNA, and in that way the aminoglycoside just prevent the formation of protein synthesis. While if I talk about the tetracycline, it's also a drug. So the tetracycline prevents the binding of the tRNA to the ribosomal subunit, and in this way tetracycline is used to inhibit the protein synthesis. And I'm going to show you this in written. For example, if you can see over here, so the aminoglycoside binds to the A side of 30 ribosomal subunit, and in this way it prevents formation of initiation complex, and it also causes misreading of mRNA. Now for tetracycline, I have the trick: tRNA tries but can't. What tRNA tries? tRNA tries to bind with the ribosome but can't, means it can't bind to the ribosomal subunit. Why? Because means the drug tetracycline inhibit the tRNA from binding it to the ribosomal subunit. Okay, now chloramphenicol, I just told you that the P on this chloramphenicol is the only drug that contain the P. So P and peptidyl transferase, so it inhibits the peptidyl transferase, and in this way inhibit protein synthesis. Macrolides, from macrolides prevents the movement of ribosomes, so thus macrolides prevent translocation. Clindamycin makes ribosome cling to mRNA, thus also it prevents translocation. And linezolid, like aminoglycoside, prevent initiation, LID. Okay, so this was all from my side. If you guys like this video, must subscribe to my channel and click the like button. Allah Hafiz for now.